Hey,
All you, Jesse. Thank you.
cool. So, good afternoon, everyone. My name is Jesse, and I am with Exploring by the Seat of Your Pants. For those who don't know, we're all about bringing conservation, adventure, and science into classrooms around the world. Today, we're doing a bit of all three in one, which is great. One big note for all the classes joining us: this is our World Oceans Day week-long celebration. In the lead up to World Oceans Day on Friday, we are doing 50 plus hangouts with amazing explorers, researchers from around the world. So thanks so much for being a part of our celebration. Right now, we've got a whole bunch of classes joining us from across North America. So I'll give them a chance to do a bit of a shout out, and then we'll introduce our speakers. So we've got Miss Wafer and her group from around the world in Ojai, California. Thank you, Wafer. Hi.
Hello, virtually, kids online school. Yep.
Awesome. We've got Mr. Rotman's grade twos in San Antonio, Texas.
Don't say hi.
We've got Miss Dillon's grade 5-6s in Farmington, Missouri.
Hello.
There are a lot of you in that classroom. Awesome. We've got Miss Davenport's 6th through 9th middle school in Athens, Georgia.
Hey.
And I think we've had everyone else join us. We've had Mrs. Winningear's grade 2s in Virginia Beach, Virginia. Hi, guys. Hi. And we'll get that mic going. And then we have Mrs. Dwinion's grade 5s in Bowmanville, Ontario. Where they're hiding. There we go. There they are. Awesome. Of course, the reason you guys are all here today is for our speaker. We're joined live from the middle of the ocean by the RV Falkor, an amazing research vessel that's going to the edge of the map to do robot exploration and study all sorts of amazing things in the ocean. We've got them live, as well as Logan Mack from the Schmidt Ocean Institute, who's able to talk about the whole shebang. So without further ado, we'll take you to the middle of the sea to hear what they're doing on the Falkor. Thank you so much for joining us, guys, today.
No more of that. Just wait.
Yep, you're good to go if you're.
Hi.
Hi.
How are you?
Good.
Oh.
If you guys are ready, yeah, go right ahead.
Introduce yourselves. Okay, so hi everybody. It's a pleasure to be here, to be in contact with so many schools at the same time. So my name is Katerina. I'm part of the biological and biochemical team of this campaign. We are doing many interesting things here on board. So during this interview, we will let you know what we are actually doing here.
Nice to meet you guys. I'm Maria Paula. I'm part of biological team too. And okay, now we are here surfing in the middle of the Pacific. And yes, we are open to interact with you.
Say where you're from.
I'm from Italy, and I work in Porto with Caterina.
Portugal. Yeah, I am from Portugal. I'm Portuguese. You guys both say a little bit about what you're studying, just to give them a context and a question to have. Okay, so we can now say something about our scientific activities. So I work I'm studying the invisible world. So the microbial communities that are floating in the ocean. So the diversity of these organisms is huge. And we know that because right now we have actually technology that allow us to look at this invisible world. So we try to understand the diversity of these organisms and what are they doing in our planet. Here in Falkor, in this campaign, the main focus is to try to target the ocean front. So the ocean front is a place in the ocean where two different mass of waters are actually encountering together. So here we are in the middle of the Pacific and in the middle of the subtropical front. This subtropical front, we have the polar waters that coming from the south, from the north, They are cooler and they have lower salinity. And these waters are just crashing with the South Pacific waters that have higher salinity, and the temperature is also higher. So in these areas, we will find like a change in water circulation and redistribution of nutrients, which is the food for the primary producers, to the organisms, to the microalgae, and many other organisms that actually are responsible to produce the oxygen that we are breathing. So these areas are really important to study and to know where are they and to map. And here in this campaign, we are just putting together many disciplines like engineering, biology, physical oceanography, to try to have a very precise information about this subtropical front. So.
Can you tell us, you got some really interesting, screens behind you with lots of information. What are those screens telling you guys?
One of the screens behind you.
Okay, so yeah. So actually, this screen here shows the data that we collect already. So the blue colors show the lower salinity at the surface, and the red colors show is the higher salinity at the surface.
What does salinity mean?
So the salinity means the amount of salts that we have in the water. So when we are eating, we can put a little bit of salt in our steak. So that makes the steak more tasty. But if you put a lot of salt, it's too much. So this is actually the same here. So we have like a tasty, nice tasty water in the north with lower salt, and and in the south the water is with too much salt, with more salt. So here is the front, is the area where the less salty water will crash with the higher salty water. So the less salty water is the blue, and the higher salty water is the red. So we are actually studying this frontier. Yeah.
Pretty cool. So the Falkor, I said that you're in the middle of the ocean, and you mentioned the subtropical front. So what's the closest land mass? Like where did you set sail from? Where are you now?
Yeah, so we left from San Diego. We are around 1,000 miles away the coast. So yeah, so right in the middle of the ocean.
Cool.
between California coast and Hawaii.
Lucky you. All right, so you mentioned that there's all these disciplines on board the ship, all these different researchers. How many people, how big is the staff doing the science on the Falkor?
Okay, we are 22 scientists, and the rest is the crew that are great, that are helping us in this research. In total, we are 42 people on board. And the boat is amazing, and we are now in the library of the boat where we are having this conference. And you can see here we have a daily meeting. We spend the most of the time together, studying, having meeting, and also chatting the night. And it's okay. Yes, I can say that is an amazing experience for me, and I feel lucky to be here and to share with you this experience.
Now, you mentioned that the boat is amazing. What makes the boat so amazing? Like, how big is it? What's going on? I mean, you have your own library, which is very cool, but what else for the kids that they would love to know?
Yeah, in terms of technology, this is, you can find different things, different instruments that are helping us in this research, starting from the CTD, that is like a probe. Imagine a big sensor that contains other sensors that are capable to register all these features of salinity that Katerina was telling you. and not just salinity, but also chlorophyll. That means the fluorescence that is in the green fluorescence that is in the water. That is an indirect way to understand how much phytoplankton there is in the water. This is the rosette that now is empty, but normally contains also bottles that when we are collecting waters at different depth with this instrument. And here is the CTD, that is the probe that I was talking before about. And CTD means conductivity, temperature, and depth. So we are able to open the bottles that are fixed in this normally in this rosette, and at different depth. So it's a kind of remote controlling, and they are going to opening at the depth that we want to sample in the front. Imagine that we would like to go at till 500 meters. So we are able to go with this till 500 meters, then to stop at the level of the higher productivity of phytoplankton, and we can detect this because there is this probe of the chlorophyll. And these are the bottles, yes, thanks, that are called Niskin bottles. And we are able from the control room, that is another room that is in the dry lab, to send this command and to let this bottle open to collect the water that we are filtering on board. So there is a wet lab where we do filtration. I mean, there is a lot of stuff that...
Excellent, guys. And so how long are you going to be at sea? How long is your journey?
We are coming back in 17th of June, but you have to consider that we need 3 days to reach the front, so 3 days also to coming back.
So altogether,
It's 3 weeks of cruise.
So I hope you like all your crewmates.
Yeah.
Are you guys okay going straight to questions with the classes now so that they can ask more things?
Let me show you some photos of some of the robots that we've been using real quick, and let the researchers talk about that, and then we can jump into questions if that's all right.
Thanks, Logan.
All right. So do you guys want to talk about what's on the screen right now? Where'd it go? Sorry, let's try that again. There we go. Could you guys tell us a little bit about what's happening here?
Okay, so here it is a rosette. The rosette is ready to sample. So down here, okay, so here is the rosette ready to sample. So the rosette, this one is with 24 bottles. So when we deploy this rosette, we collect samples until 500 meters, and we open 4 bottles at each depth. So we collect 36 liters of water at each depth. So here is when the rosette is up. So that's the time of collecting the samples. So here I am collecting samples to do nutrients. So this will give us information about the chemical quality of the water. That's very important because it's the chemistry that will drive the distribution of the biological biomass. Oh, here is the sonar, so I can show this one. I don't know. So here, this system will actually give us areas of where we can have biomass, biological biological cells floating in the water. So here we can see that in this area, the blue is a little bit brighter.
so he actually is showing a CTV. We're not seeing it because Jesse, can you get on that screen? Here. Logan's still showing a photo there. Wow, so he's showing it there.
Sorry, guys.
Okay, so these are the 1, 2, 3, 4, 5, 6, 7 underwater vehicles. So they are autonomous vehicles. So we'll be able to, the engineering team, what they're doing is that they are deploying these systems in the water, and then they will be able to drive alone through a certain mission that, and during that period that they are in the water, they goes down, and then they goes up, and then they goes down, and then they goes up. And every time they goes up, they will send the data that they collect to the ship. So then we will know what was the temperature, the depth, and the salinity, where, I mean, in that area where these systems were operating. So then there are also some drones. So these drones are also flying across the front, and they have some cameras, and these cameras actually can capture some information about the temperature of the water.
Yes.
And also they also have some sensors that will capture, I mean, some information about areas of biomass. So they have some sensors of DMS. It's a compound that is produced when we have a lot of phytoplankton production, algae production. So when we have a lot of algae, we have a lot of production of this gas, and this sensor will be able to capture this gas, to sniff this gas. Okay, so here is. So the engineering team was deploying underwater vehicle, autonomous vehicles. So that's the way that we have to do. So we have to go to the small ship, and then with the small ship, we'll be able to deploy it and also to collect these underwater autonomous systems. Yeah, and here is all the team, almost all the team, all together with all the instruments that we are actually working on board. So yeah, so this picture was Mary that just took yesterday with us. So, and here again, so the picture with the drones and the underwater autonomous vehicles.
Great. Thank you guys very much. I think it'd be great to start questions now if possible.
All right. So we'll start with Miss Waver's group. So if you have a question to pass along from one of your students.
I do. And you can hear me, I'm guessing?
Yep.
Awesome. So we have a student in Cypress, Texas, Davin. He's in 8th grade, and he's wondering about how the Rosettas are made.
Plastic.
Rosette.
Yeah.
Explaining metal, the structure.
The structure of the rosette, yes.
Okay.
It's imagine the rosette is the shape, so it's made by metal, and as you can see, okay, it's okay. It's a metal structure, and inside the Niskin rosette, the Niskin bottle, sorry, are made by strong plastic, and yes, and here there is the CTV. So mainly is metal, yes.
Quick note before we go on to our second class, we've got 20 groups watching on YouTube live. If you guys want to send in your questions in the chat bar on the right, I can pass along a few as we go along. So just do keep that in mind if you want to send in questions. But first we'll go to Mr. Rotman's class. You guys have a question. You might have to demute your own microphone. It's not letting me do it from here. So just a little microphone symbol top of your screen and you should be good. Yep, you're good.
All right, go ahead.
How long have you been on the water?
Sorry, start again.
How long have you been on the water?
Okay, hi. we are here since the 28th of May, actually, because we need 2 days to familiarize with boat, with all the safety stuff that we have to learn to stay on board, to organize all the stuff to leave. So from the 28th of May to the 17th of June. So let's say 21 days, more or less.
Thank you.
All right. We'll go to Miss Dillon's class. If you guys have a question.
What kind of scientist are you, and what have you found out about the sea?
Nobody can be talking. Okay, go ahead.
Yep, you're good.
What kind of scientist are you, and what have you found out about the sea?
Okay, thanks for your question. I am a biologist too, and I would like to look for this invisible world too. So the microbes, the marine microbes, and actually here you have this great challenge to see it. Nevertheless, it's invisible because you are very close to it. So we are filtering water to capture this marine microbial world. So I'm investigating these microbes because I think that are very important. Nevertheless, they are invisible because they are the basis of our life. Because they produce oxygen and they are the basis of the food webs. And I think that this ecology, marine ecology, is very important to understand and to improve our knowledge in this field for our life.
Okay, so microbes. Do you guys know what
they are?
Okay, put your hands down.
I
I don't know what they are.
We can stick with that. That's fine. Okay, we'll go to Miss Davenport's class. If you guys have a question, again, come on up and go for it.
Okay, all right. Okay. I was wondering what the food is like on the ship.
Okay, which kind of food? So the food is fantastic on the ship. So we have a very, very good cooker. So we actually have 3 main meals on the ship. The breakfast, that will be between 7 a.m. to 8 a.m. So then we have our lunch. It's a little bit early. We start our lunch at 11:30 until 12:30. And then we have a dinner between 5:30 to 6:30. And we have a lot of salads. We have a lot of different foods, soup. Yesterday, for example, we had a Mexican dinner. It was really good with tacos and guacamole and all these nice things. Sometimes we have cake when we have a birthday. Maria Paula had a birthday in the first day, so we had like a cheesecake. It was really nice. So actually, it's very, very good, the food on board. I think everybody will agree with that.
I have to ask a quick follow-up. Do you have seafood on the ship?
Yes.
Yes. Oh, yes. We have
Okay.
So, yeah. Yesterday we had shrimp. And we have white fish. We already had white fish. Yes. Very often.
Gourmet food. All right. Let's go to Bowmanville.
Although we didn't fish yet, any fish.
Okay. All brought on board with you. Perfect. Let's go to Bowmanville. If you guys want to demute your own mic, again, little symbol top of your screen, and you'll be good to go. There you go. Yep. Hi. So we have you, like your mic is demuted, but we can't hear you for some reason. I don't know if there's a volume or anything. Sorry. Worst case, while you're trying to figure that out, there's a little blue square top left of your screen, and you can type in a question there. Yeah. So the question is, did you ever get seasick? Do you ever get seasick?
Yes, seasick. Oh, yeah. So in the beginning, we have, I think we almost all experience seasick. The weather was not very good by the time we were in transit to the area of study. So we had 3 days with big waves. Let's see, 3 meters, something like that. But of course, the team were not used to the sea, so we were feeling very tired. So we slept more. So we try also, since we were in transit, so we were not doing too many things to get our body get used to the sea. But after that, I think after those 3 days, everybody starts recovering, and right now I think everybody is doing well.
Okay. Here. Question online. Oh, sorry guys. Question from a group online. How deep does your equipment go? You talk about robotic vehicles going up and down in the water. How low do they go?
Yes, nevertheless, we are sampling at the 500 meters. We are with the rosette. We arrive at we arrive at 100 meters. But with the AUVs, we have an engineer here, Joao, that can tell you more about the technology because we are just biologists.
Hello. Our AUVs, they go up to 100 meters depth. Most of them.
How many feet is that? Those are American students.
Seats. Oh, no sorry.
Can you?
We will let you know the seats in a moment.
Okay. All of them, they are equipped with the CTD, so the sensor that gives the salinity of the waters. And some of them have special instruments like chlorophyll, turbidity, some instruments and some probes to measure the pH of the water also. Yes, most of them, they can run up to 60 hours, so 60 hours straight without charging. And well, 1 or 2, they have more instruments, so they withstand less time on the water, so they can go up to 34 hours.
Excellent. Thank you so much. All right, we'll go back to Miss Waver's group if you guys have a second question.
We do, and I just want to let you know we're a virtual school, so if you see me going back and forth, I have my students in another room, and so I'm looking at their questions, and we're just loving the wake. We were all commenting, seeing the wake of the ship is really exciting for us. It makes us feel like we're there. So Amaya is a 7th grade student, and he is in Southern California, and he's wondering if the Falkor uses renewable energy sources like solar or wind power for their energy.
We have some solar. We have some solar, but not.
Well, I was told that Falkor has some solar winds, but solar energy, but most of the energy, no, it's produced by petrol, right? Yeah.
Okay, excellent. Let's head back to Mr. Rotman's class. You guys have a second question. Just, yeah, come on up, mute the mic, and we'll be good to go.
Okay, let me see if we have any second questions. Am I good?
Yep, you're good.
Okay. All right, what's your question? Okay. There we go. All right, go ahead.
Yep.
Do you have fish on the ship?
We catch fish.
No, we don't do fishing on the ship. We fish AUVs when they are on the water and we want to recover them, and also the rosette, but we don't fish any animals.
Okay.
Thank you.
Good question, though.
I'm afraid I'm going to have to jump in. We're running really short on time. We've got about a minute left. If there are questions moving forward, we can probably handle one more. I just also wanted to let you know that we are available online at schmidtocean.org, and we're also on Facebook and Twitter and Instagram. So if you have any questions, you can go on social media or go on our website and send us some emails. We can pass them along to the researchers. Jesse, if you want to take one more question, I think that's what we can do.
Sure, yeah. Well, let's do a quick one from Miss Dillon's class then. Go right ahead.
What do you want to learn about the microbes you study?
Good question.
Hi, thank you for your question. Very good question. So what you want to learn? Well, here in this specific campaign, so as you remember, I just said a little bit some time ago that we are trying to study an area where we have a gradient of different water masses. So, and what you want to know is how these microbes are distributed across these water masses, and what are they doing across these water masses. So what we're going to do is that we are like Maria Paula just said, we concentrate all the cells that are floating in 5 liters of water. Then we extract or isolate the DNA. It's like the bark, and then we will have like a barcode to use to identify the different species that we have in our sample. And with that, we will know that if the species are different from one area to the other area of the front. We also will be able to know what are the functions of these organisms, what they are doing. Are they fixing carbon dioxide? Are they producing a lot of oxygen? You know, these kind of functions.
Outstanding. All right, Logan, so before you stop the broadcast, what we always do, we're going to mute every class's microphone. If you guys want to join us and say a big thank you to the Falkor team. So thank you so much, guys, for joining us today. Thank you.
Thank you. Thank you.
Thank you. Thank you.
Awesome. We look forward to having you guys back soon. We have lots more hangouts throughout the week, and we look forward to having the Falkor join us further on the journey. Thanks so much, guys.
Guys,
Have a nice day, everyone.